Hi, everyone. I'm Siyu. I want to thank the organizers for this opportunity. And this is my first conference talk as a PhD student. As we all know, understanding protein function is important. Determining protein functions experimentally is challenging. So computational approaches are needed. Traditional approaches use different scales of data, and I will talk more about this. First, each protein is made up of a sequence of amino acids, so it is obvious to predict functions directly from their sequence. The pro is that we have a large amount of sequence data. However, proteins with dissimilar sequences may have similar functions. For example, there are two proteins named Safrina1 and Safrina3, which have different sequences but share the same cellular roles. Why that happened? It is because they have similar structures. For both of them bind to almost the same binding surface of another protein called eline. Clearly, the amino acids of a protein fold into a 3D structure, and it is the 3D structure that directs what other proteins a given protein can interact with and what functions it can perform. So using 3D structure to predict function is quite promising. The drawback is that there is less experimental 3D data, structure data compared to sequence data. However, this is not a major concern now with methods like AlphaFold. Moreover, proteins often don't function independently because they typically need to interact with other proteins through protein complexes to perform their biological roles. These complexes can be modeled as protein-protein interaction, or PPI. When we consider all PPIs in a cell, a PPI network can be formed. In such a network, nodes are proteins and edges are PPIs. The physical bindings between proteins should be efficient in predicting protein function. However, PPI data are incomplete and noisy. Well, modeling interactions between proteins as a network is straightforward. What might be less obvious is that the 3D structure of a protein can also be modeled as a network. Specifically, it can be modeled as a protein structure network, or PSN, where nodes are amino acids, edges drawing amino acids close in 3D space. Unlike the traditional approach of using only PSN or only PPI networks to predict function, our lab recently proposed a method to integrate both of them together. The networks at different levels are integrated through a network of networks framework, aka NON. Each protein can be modeled as a node in a PPI network. It can also be modeled as a PSN at a lower level. When predicting function, we use Go terms by predicting whether a protein is annotated by a given Go term or not. NON is powerful. For 30% of the Go terms, only NON approaches made non-random predictions. And it also outperformed PSN alone and PPI network alone approaches in about half of the Go terms. However, it is only half. In the other half, the traditional single-level approaches were superior to the NON framework. So this data integration did help, but did not always help. And I'm interested in seeing whether this can be improved as follows. Until recently, final 3D structures were modeled as static PSNs. However, protein folding is a dynamic process, with certain parts folding before others, as this figure shows. To try to capture this dynamic process, our lab recently introduced dynamic PSNs. A dynamic PSN consists of multiple PSN snapshots as visualized. Dynamic PSNs significantly outperform static PSNs in the task of protein structure classification, which is about predicting the folding type of a protein, such as scalp or calf. They also work well in predicting function structure change upon ligand binding. This is our newest work, and Aidan Wells will talk more about this project at the 3D Cozy tomorrow afternoon. Importantly, Static PSNs have already been shown to perform better than traditional methods, such as sequence or 3D structure analysis in the task of protein structure classification. As dynamic PSN works well in some other tasks, we want to ask, will modeling protein 3D structure as dynamic PSNs benefit protein function prediction? In the first context of analyzing PSNs only, we previously used dynamic PSNs in other tasks, and our contribution in this work is using dynamic PSNs in protein function prediction task. In the second context of analyzing PSNs within the NON framework, we have previously used an NON with static PSNs in protein function prediction. And in this work, we use an NON with dynamic PSNs in the same task. For data, we use a human PPI network from BioGrid. We generate both static PSNs and dynamic PSNs for all proteins from the PPI network with 3D structures in PDV. And we use gene ontology terms as labels for this binary classification. For each of the 3 network types, we extract the corresponding graphlets. Graphlets are Lego-like building blocks of a complex network. And it has been proved that it is a powerful tool for analyzing complex networks. For each network type, we have its corresponding single level approach as shown in the red arrows. Additionally, we combine the features across the levels to create the static NON and dynamic NON approach. Then we perform the standard 5-fold cross-validation as well as logistic regression, and then we evaluate the result with 4 measures. For the results, when comparing static versus dynamic PSNs at the PSN level only, we count the number of goal terms for which a given approach is superior to the other. In this chart, x-axis shows the 4 measures and y shows the number of goal terms. With static being superior in red and dynamic being superior in blue, the deeper the color is, the higher the extent of superiority is. And in each bar, the non-random results are marked with lines. A surprising finding from this figure is that both approaches predict random results for most goal terms. When we focus on non-random results, static PSNs are better than dynamic PSNs in 3 goal terms, while dynamic PSNs outperform static PSNs in 30 goal terms. To focus on results with absolute superiority, rather than just minor difference, we set a superiority threshold of at least 10%. Among these, we find that static PSNs are superior in at least 1 measure for a total of 2 goal terms, with none of them supported by all 4 measures. Dynamic PSNs, however, are better in total of 28 goal terms, with 8 supported by all 4 measures. When we dig deeper into these 8 goal terms, we find that half of them contains the phrase phosphorylation in their name. This ratio is much higher than 9% observed in the entire dataset. This indicates that although no single approach works well for all goal terms, there might be a certain approach, in this case dynamic PSNs, that perform well for specific biological process. And we are examining whether it has any meaning biologically. When we're analyzing static PSN versus dynamic PSN within the NON framework, we have a similar type of figure, but this time comparing static NON to dynamic NON. There are still many random results for both approaches. Of the non-random results, we find that the static and dynamic approaches are more comparable to each other because they are superior for a similar number of goal terms. According to the Venn diagram with the superiority threshold, we find that both approaches bring value, with 22 goal terms here and 16 goal terms here. Static NON is superior in more goal terms than dynamic NON. I have talked about comparing static PSN to dynamic PSN, and also comparing static NON to dynamic NON, but that is not the end. We also want to compare all static approaches versus all dynamic approaches, by grouping the full approaches into two categories. In other words, we want to ask, in how many Go terms, at least one static approach is non-random and better than both dynamic approaches. The result is 27 Go terms for at least one measure. We also want to know the result where at least one dynamic approach is non-random and better than both static approaches. The result is also 27. When we set the superiority threshold to at least 10%, we find that static approaches are better than dynamic approaches in total of 21 Go terms. For the 5 Go terms supported by all 4 measures, it is always the static NON that is superior, never static PSN, which shows the importance of considering NON. Dynamic approach are better than static approaches in total of 17 Go terms, with 6 of them supported by all 4 measures. It is either the dynamic NON, dynamic PSN, or both of them that are superior. Remember, the PPI network approach is also a static approach. Now we want to compare all 3 static approaches versus the 2 dynamic approaches, which means the first question will remain the same. But for the second question here, it will be changed from both static approaches to all 3 static approaches. And here's how the results change. Static approaches are better in 49 Go terms, while dynamic approaches are better in 26 Go terms. With the superiority threshold applied, the result adjusts to a total of 38 Go terms for static approaches and total of 15 Go terms for dynamic approaches. The changes from 27 to 26, 17 to 15, and 6 to 5 indicates that PPI network approach does not diminish the effectiveness of the dynamic approaches on the Go terms where the dynamic approaches works best. The changes from 27 to 49, 21 to 38, and 5 to 16 indicates that PPI network approach adds value to new Go terms, where neither dynamic approaches nor the other 2 static approaches work effectively. In other words, even when considering the PPI network approach, dynamic PSNs, dynamic NONs, and static NON continue to demonstrate their value. Finally, we can conclude that dynamic PSNs add some value to all analyses, although they still make random prediction for most Go terms. Secondly, the network of network framework also brings value, no matter if it is static NON or dynamic NON. Third one, no single approach works well universally, not even for the majority of Go terms. For example, with respect to AUPR, there are 37% of all analyzed Go terms where none of the 5 approaches work, meaning all of them failed. When considering non-random results, the approach that works best for most Go terms, which is the PPI network approach, still works best for only 28% Go terms. Why do PSNs provide less predictive power than PPIs? Recall that in other 2 tasks, we have shown that PSNs, especially dynamic PSNs, capture the 3D structure of proteins extremely well. If they capture the 3D structures, why they are not linked well to function, or at least not as well as a PPI network approach? Is this a data problem? How much can we rely on the Go term data we use as a label? Or is this due to just the application? When we correct the multiple hypothesis testing, we lose a lot of statistical significance. This is a shock, but I hope you don't feel like you wasted 20 minutes. This could happen because none of the results make sense, but we don't believe that is the case. Here, it might be due to the nature of the analysis we are conducting. Primarily, we have many approaches, and we analyze each approach for each individual Go terms across 4 measures. According to our results, although no approach works universally, it is possible that each approach works well for certain biological process. However, in order to find out which ones those are, we have to test the significance of the approaches on all of the Go terms. But when we do that, we will lose lots of these significant signals. Is there a need for new methodological improvements? We find that there are existing NON studies, some of which have advanced methodological ideas and integrate deep learning components. Can we benefit from their methods in our application? Noted that almost all of these studies, they focus on different data types, different network analysis tasks, and different biological applications. It is not trivial to apply one method to another task and application, and vice versa. Additionally, even if we could do that, it is often impractical because these studies, besides our work, typically don't provide their code and their data. As the NON topic continues to gain importance, one future direction that needs attention is the development of an evaluation framework for different existing studies, as it is impossible to compare them with each other currently. This challenge in the field of network biology, along with many other challenges, is discussed in our lab's recent perspective paper, as illustrated here. And finally, thank you, and I'm happy to take any questions.
Is there any question? Any online question? No. Any question from the audience?
If you just do a sequence similarity search like BLAST, you will outperform your method.
I'm sorry. Can you repeat your question?
So for a subset of 107 GOTAMs you look at, if you just perform BLAST search of your test protein through your training data set, how will it perform?
We don't know that. We didn't test, but since we are like network person, and we try to focus on like network company values. But I find there are a lot of work they do on text with a name.
I think like Kappa people here, and we think BLAST is the necessary baseline. So maybe we need to do that comparison.
Okay.
Okay, so do we have a question? I have just a very, very small one. Regarding the information content of the Go terms that you are predicting, how deep are they? What is the level of detailed information that you can extract from these Go terms? Are they very general? Are they very specific?
We find like all of the 107 Go terms related to the data we use, and we find that we try to find some relationship between the size, the level of the Go terms in the tree, but not that much significant result we find right now. But maybe we need more bio-related information to find more significant things.
Okay, so thank you. Let's applaud the speaker. And now